into the electron transport chain. Let's really quick account for what's happened so far. We have, this is not balanced, I'm just going to write it out. At this point, through glycolysis and the Krebs cycle, all of the carbons and oxygens from glucose are gone. They have now been converted to CO2 and you exhale them as a waste product. So the carbons and the oxygens are gone. We have not used this oxygen yet. Oxygen has not been necessary at all for the reaction so far. The electron transport chain is where the oxygen, this oxygen, is going to come in. So make sure that you know, because it's a question I guarantee you would be asked, where do the oxygens for CO2 come from? They're the ones that are coming from glucose. Where do the oxygens that form water come from? They're the ones that are going to come from the oxygen that you inhale. So that is going to be the job of this oxygen. It's going to pick up hydrogens at the end. Now, where are the hydrogens right now at this point? Where are the hydrogens? I heard somebody say it. Something picked them up. Coenzymes. Not acetyl-CoA, but what are the other coenzymes we talked about?
Ooh,
3 letters. NAD is one of them.
NAD. FAD.
And FAD is the other one. So all of the hydrogens, if we add this up, at this point, whoops, we've made 10 NADHs, and we've made 2 FADH2s. That means that we've got 10 hydrogens here, and 2 hydrogens because FADH2 came from, it was already FADH, just so you know. So it only took 1 hydrogen from glucose. If you add that up, we get 12 hydrogens. There's all 12 of our hydrogens from glucose. And that's all that's left to now feed into the electron transport chain. Just the hydrogens. How many ATP have we made so far? Not more than 2.
4.
We've made 4. We made 2 in glycolysis, and we made 2 in the Krebs cycle. So the electron transport chain is going to use only hydrogens and oxygen that we inhale at the end, and it's going to generate an additional 32 ATP. And so it's obviously going to be a very efficient process. So I had you guys write this a minute ago. So let's go ahead here. So what is the electron transport chain? What it actually is, they're calling it a chain. It's a bunch of proteins in a row on the inner membrane of the mitochondria. Going back really, really quick, whoops, drawing a mitochondria. What was the middle called where the Krebs cycle occurred? Good, that's the matrix. And now what's going to happen is these are going to be little proteins literally right here in this membrane. And what they're going to do is they're going to take the electrons off of NADH, basically the hydrogens are going to come off of NADH and FADH2. FADH2 will go back to being FADH, so it'll just lose 1 hydrogen. And they're going to pass those to oxygen. But here's the thing. The hydrogens and the electrons are not going to travel together. Hydrogen has 1 electron. What's going to happen is, and it's going to tell you this in this last step down at the bottom, but in other words, you have NADH, the H is going to break off, and the electron from hydrogen is going to go down the electron transport chain. In other words, this is a hydrogen ion. It's missing its electron. The hydrogens are not carried in the electron transport chain. Electrons are carried in the electron transport chain. And I'll show you an animation in a minute that kind of illustrates what happens. But in general, what's going to happen is the hydrogens are all going to get dumped off in the inner membrane space. So if this was our mitochondria, our hydrogens are all going to be sitting out here in this space. The electrons are going to go down the electron transport chain. And at the end of the electron transport chain, the hydrogens and the electrons will both hook up with oxygen and we'll end up with water. So this is called the electron transport chain because it's carrying electrons. It's called oxidative or chemiosmotic phosphorylation because it has to do with redox reactions. Because what's happening is oxygen's at the end, and it's the most electronegative thing in the chain. And it's sort of sucking all the electrons towards it. It's got such a strong pull that every time a protein grabs it, the next protein, it's more attracted to the next protein in the chain, and so on until they get to oxygen at the end. And so little bits of energy are basically going to be released in this process. And this is what I was just saying. So the hydrogen ions are going to get dumped off in the intermembrane space, the space between the inner membrane and the outer membrane. Only the electrons are going to get passed through the proteins. And I have a diagram of this, which I'll show you in a second. All right, so there's more. What's going to happen is we're going to end up with more hydrogens on one side than the other. The hydrogens all build up, and they come back in through an enzyme called ATP synthase. It sounds almost like it's too easy to be the answer. What if I was to ask you what actually makes the ATP? An enzyme called ATP synthase. Literally ends in ACE, tells you what it does. Synthesizes ATP. Not a trick question. That's actually what makes the ATP. But what happens is the hydrogens flow through that enzyme, and when they do that, that's what's going to generate the ATP. Now, the old way we made ATP, the four that were made prior to the electron transport chain, they were made by what we called substrate level phosphorylation, where literally there was an enzyme and it pulled a phosphate off of something and it stuck it onto ADP. And it was very slow and it only made a couple ATP. This is not like that. What's going to happen is as hydrogens flow through this enzyme, we're going to generate multiple ATP. And this little section at the bottom here is really the key to the whole thing. If you don't understand anything else on this slide, this last thing here is the most important thing of all. So every time an NADH goes through the electron transport chain and gets rid of its hydrogen, we generate 3 ATPs. So it's not a one-for-one thing. And every time an FADH2 goes through the electron transport chain and gets recycled, it generates 2 ATPs. So this is where all those ATP are going to come from. The NADH and the FADH getting rid of their hydrogens is going to generate large numbers of ATP for every hydrogen that breaks off. So this is going to make a lot more ATP, and this is called chemiosmotic phosphorylation or oxidative phosphorylation. Those two words refer to the fact that, number one, it involves redox reactions, where electrons are getting passed along, and it involves a gradient across a membrane, chemiosmosis, a buildup of chemicals and charges on one side of the membrane compared to the other. But bottom line, if you don't understand all the details, if you know this last line, this, you can answer most of the questions. This line and the name of the enzyme. Honestly, the rest of it, maybe there's a question somewhere, but a majority of it can be answered using just that information. I could ask you a question. What kind of application could I ask you about this? I could say, you start with 4 NADHs in the electron transport chain. How many ATP will you be able to make? 12, exactly. If every NADH makes 3, simple multiplication, 4 times 3 is 12. That's the kind of application question you could be asked. So if you know just this one little thing, and the fact that the enzyme is what makes it, you should be in good shape. So this is going to summarize everything. So going all the way back, we started with glycolysis. ETC, in this case, is referring to the electron transport chain. This is electron transport. So glycolysis made 2 ATP net on the spot in the cytoplasm. Glycolysis also made 2 NADHs. Now, this is the reason why the video, at least I think this is why the video was saying that this electron transport chain made 34 ATP instead of 32. Glycolysis happened in the cytoplasm. The electron transport chain happens in the mitochondria. It turns out that these 2 NADHs in the cytoplasm cannot leave the cytoplasm to go into the mitochondria. So even though technically I told you every NADH can make 3 ATP, this doesn't end up making 6 because the NADHs actually give their hydrogens to FADH. That's what then goes into the mitochondria, and every FADH can only make 2 ATP. So I think that's where they were getting 34 instead of 32, is they were ignoring the fact that these 2 NADHs, when they get recycled, they go back to glycolysis again. They don't leave the cytoplasm. The rest of this does add up correctly, though. So the next thing that happened was the oxidation of pyruvate.
Can I follow the scarf thing?
Yeah, the oxidation of pyruvate made 2 NADHs also. Again, in the electron transport chain, those will now generate 6 ATP. We also made 2 CO2s. The carbon dioxide is a waste product, so that we exhale that. That's the carbon dioxide. That's getting rid of the carbons from glucose. We're done with those. Our next step was the citric acid cycle. It made 4 more CO2. There's all your carbons from glucose have become carbon dioxide. You exhale it. It's a waste product. Interesting little tip, photosynthesis uses CO2, takes the carbons, puts them back into glucose again. So photosynthesis is actually taking the carbon we exhale, putting it back into glucose. You could almost imagine eating the same carbon you exhaled the day before. Pretty weird. Whoa. I know. All right. It also made 2 ATP on the spot. That was our 4 ATP that were made without the electron transport chain. Then it made 6 NADHs, and it made 2 FADH2s. When you add up all these ATP that are made just in the electron transport chain, we get 32 ATP. That may not match your notes because I added another line here. Then when we add the 4 that were already made, the 2 that were made in the citric acid cycle and the 2 that were already made in glycolysis, adding those in, that's where we get our total of 36. So that pretty much summarizes the entire long, crazy process that we've gone through. So glycolysis made 2 on the spot. If there was no oxygen, it would have gone to fermentation. You wouldn't have made any more. That would have been it. You make 2, you make lactic acid. Or if you're a bacteria, you make alcohol. If oxygen is present, this continues. Goes to oxidation of pyruvate, which does not make any ATP on the spot, but does make NADH. We go to the citric acid cycle, which makes 2 ATP on the spot, and then a bunch more NADH and FADH. And then in the electron transport chain, all those NADHs and FADH2s give away their hydrogens, and that's what ends up making the ATP. I'll wait.
Wait, so